Hello, welcome to Module 5. This is Education Lesson. My name is Wesley Martin, your lecturer. Make sure before you begin to subscribe so that way you can follow along for all content. Hopefully be releasing every few days. Today we're connecting two systems that, at first, seem unrelated. How we sense the world and how we move through it. The link between them is a single idea: transduction. The conversion of one kind of signal into another. A sensory receptor turns a physical stimulus, light, pressure, a chemical, into an electrical signal the brain can read. A muscle does the reverse. It takes an electrical command and turns it into mechanical force. Over the next 20 slides, we'll follow that thread from the outside in and back out again. First, how receptors detect and encode stimuli, then how skeletal muscle converts excitation into contraction. Keep that theme of signal conversion in mind. It's the spine of everything we cover today. Before we dive in, here are the 6 things you should be able to do by the end of this lecture. The first 3 are about sensation. Classify sensory receptors and explain transduction. Describe how the nervous system encodes the 4 features of a stimulus. And, jumping across to muscle, relate skeletal muscle structure to the sliding filament mechanism. The second 3 go deeper into muscle, sequence the events of excitation, contraction, coupling, with real emphasis on calcium. Explain how the body grades muscle force, and finally compare the 3 muscle types. I'd suggest coming back to this slide when you review. If you can speak to each of these 6 points in your own words, you've got the module. Let's start with the sensory side. Part 1. Sensory Physiology. Everything in this section answers one question. How does a physical event, out in the world or inside your body, become a signal your nervous system can actually use? We'll build up in order. What the receptors are, how they perform that conversion, and then how the resulting signals carry meaningful information about the stimulus. Take a breath here, and then let's begin by sorting the receptors themselves. There are 2 independent ways to classify a sensory receptor, and every receptor gets one label from each. The first is by the type of energy it detects. Mechanoreceptors respond to physical deformation, touch, pressure, stretch, even sound waves. Thermoreceptors respond to temperature. Photoreceptors respond to light. Chemoreceptors respond to molecules, which cover taste, smell, and blood gas sensing. And nociceptors respond to tissue-damaging stimuli. That's pain. And we'll give it its own slide. The second axis is location. Exteroceptors face outward and monitor the external environment. Interoceptors sit in our organs and vessels, tracking the internal state, and proprioceptors in muscles, tendons, and joints tell us where our body is in space. So a receptor in your fingertip is a mechanoreceptor and an exteroreceptor at the same time. This is the central event of sensation. When a stimulus hits a receptor, it opens ion channels in the receptor's membrane. That's transduction, the conversion of a physical signal into an electrical one. The immediate result is a receptor potential, a graded depolarization, meaning a stronger stimulus produces a larger potential. But notice the word graded. This signal is local, and it fades with distance. So if it's large enough to reach threshold at the trigger zone, it gets recoded one more time into a train of action potentials. And here's the key. Action potentials are all or none, identical in size. So stimulus strength can't be carried by their amplitude. It's carried by their frequency. A stronger stimulus fires more action potentials per second. Hold on to that idea, because it's how intensity gets encoded. Now, a train of identical action potentials. How does the brain know what they mean? Consider 2 features first: modality and location. Modality is what kind of stimulus it is. Touch versus warmth versus light. It's coded by the labeled line principle. Each pathway is hardwired into one modality and reports to a dedicated region of the brain, no matter how it's activated. That's why pressing on your closed eye produces a flash of light. You've mechanically activated the visual line, and the brain can only read it as light. Location is coded by which receptive fields are firing. Where receptors are small and densely packed, your fingertips, your lips, you get fine two-point discrimination, the ability to tell two nearby touches apart. Where fields are large, like your back, localization is much coarser. The other two features are intensity and duration. Intensity uses two codes at once. The first is frequency coding, which we already met. A single receptor simply fires faster for a stronger stimulus. The second is population coding, or recruitment. A stronger stimulus also activates more receptors. Together, these let you distinguish a gentle touch from a hard press across an enormous range. Duration, in principle, is just how long the receptors keeps firing while the stimulus is present. But here's the twist. Receptors often stop reporting a stimulus even when it hasn't changed. Their firing rate declines over time. That phenomenon is called adaptation. And it's important enough to earn its own slide, which is exactly where we're headed next. Adaptation is a decrease in receptor response to a sustained, unchanging stimulus. It's a property of the receptor. The stimulus hasn't gone anywhere. Receptors fall into two camps: tonic, or slow adapting. Receptors keep firing the whole time the stimulus is present. They're monitors of ongoing conditions. Proprioceptors are a good example. You always need to know where your limbs are. So are nociceptors. Pain shouldn't fade while the danger's still there. Phasic or fast adapting. Receptors fire a burst at the onset, go quiet, and often fire again at offset. They signal change rather than steady state. This is why you feel your clothes when you first put them on and then stop noticing them, and why a smell fades within minutes. Adaptation is the nervous system filtering out the constant, so it can attend to what's new. Let's zoom out to the senses as a whole. Broadly, they split into two groups. Somatosensation covers the general senses: touch and pressure, temperature, pain, and proprioception. And these are distributed all over the body. The special senses, by contrast, are localized in the head and served by dedicated organs: vision, hearing, balance, taste, and smell. And here's a nice check on what we've already learned. Every one of these maps back to a receptor class from a few slides ago. Vision uses photoreceptors. Hearing and balance use mechanoreceptors. Hair cells bent by fluid movement. Taste and smell use chemoreceptors. So all this apparent variety comes down to the same handful of transduction mechanisms, just packaged into different organs. Now, one general sense deserves special attention. That is pain, nociception. Nociceptors are free nerve endings, literally bare endings in the tissue, and they're high threshold. It takes a genuinely strong, potentially damaging stimulus to activate them. Crucially, because their job is protective, they adapt little or not at all. You don't want pain to fade while the injury persists. Pain travels on two kinds of fiber, and this is why an injury often has two phases. A delta fibers are myelinated and fast, carrying the sharp, prickling, well-localized first pain. The instant you stab your toe. Stub your toe, rather. C fibers are unmyelinated and slow, carrying the dull, aching, diffuse second pain that follows and lingers. That two-fiber design is why you first feel a sharp jolt, then a throbbing ache, and that closes out sensation. Now let's turn to muscle. Part 2. Muscle Physiology. Notice we're staying on the same theme, transduction. On the sensory side, we converted a physical stimulus into an electrical signal. Now we run it in reverse. We'll take an electrical signal, an action potential, and convert it into mechanical force. We'll start with the big picture of the three muscle types, then spend most of our time on skeletal muscle, its structure, how it contracts, and how the body controls how hard it contracts, before comparing all three at the end. Quick orientation before the detail. There are three muscle types, and they differ along a few axes. Skeletal muscle is striated. It has that banded appearance under the microscope. It's under voluntary control. Its cells have many nuclei pushed to the edge, and it moves the skeleton and maintains posture. Cardiac muscle is also striated, but involuntary, with typically one central nucleus per cell, and it pumps blood. Smooth muscle is not striated, is involuntary, also has a single central nucleus, and forms the walls of hollow organs, blood vessels, gut, airways. We'll dig into skeletal muscle for most of this section, because it's where the mechanism is clearest, and then return to contrast all three at the very end. To understand contraction, we need the architecture. A whole muscle is made of muscle fibers, individual cells. Each fiber is packed with myofibrils, and each myofibril is a chain of repeating units called sarcomeres. The sarcomere is the fundamental contractile unit, and it runs from one Z-line to the next. Inside it are two kinds of filament. Thin filaments, made mostly of actin, anchor the Z-lines at each end. Thick filaments, made of myosin, sit in the center. Their arrangement gives the bands you see on screen. The A band spans the full length of the thick filaments. The I band contains only thin filaments near the Z-lines. And the H zone, in the middle, contains only thick. Keep this map in mind. The next slide is all about what happens to these bands when muscle shortens. Here's the theory that ties structure to function, and its name gives away the punchline. When a muscle contracts, the filaments do not shorten. Instead, the thin filaments slide inward, past the thick filaments, toward the center of the sarcomere. Watch what that does to the bands. The Z-lines are pulled closer together, so the whole sarcomere shortens. The I band shrinks, because the thin filaments are moving into the thick filament region. The H zone shrinks too, as thin filaments encroach on the center. But the A band, the length of the thick filaments, never changes, because the thick filaments aren't getting shorter. That constant A band is the diagnostic fingerprint of the sliding mechanism. So the obvious next question is, what actually pulls the thin filaments inward? For that, we need calcium. Excitation-contraction coupling is the bridge from the nerve signal to the sliding filaments, and calcium is the switch that makes it happen. 6 steps. 1. A motor neuron fires and triggers an action potential across the muscle fiber at the neuromuscular junction. 2. That action potential spreads down the T-tubules, invaginations that carry it deep inside the cell. 3. Voltage sensors that there open calcium release channels in the sarcoplasmic reticulum, the muscle's internal calcium store. 4. Calcium floods out into the cortisol. 5. This is the key event. Calcium binds to troponin, which drags the tropomyosin strand off the actin's binding sites. 6. With those sites now exposed, myosin can finally attach, and the cross-bridge cycle begins. Take away the calcium and every step reverses, and the muscle relaxes. Now let's look at the cross-bridge cycle itself. This is the engine that actually does the pulling, and it's a 4-step loop powered by ATP. Step 1. Attachment. An energized myosin head binds to the exposed site of actin, forming a cross-bridge. Step 2. The power stroke. The head pivots, physically dragging the thin filament inward toward the center, releasing its ADP and phosphate as it does. Step 3. Detachment. A fresh ATP molecule binds to the myosin head, which breaks it away from actin. Notice ATP is required to let go, which is exactly why muscles stiffen in rigor mortis when ATP runs out. Step 4. Recocking. The myosin hydrolyzes that ATP, using the energy to swing back to its ready position, and then it repeats. As long as calcium keeps the sites exposed and ATP is available, the cycle turns over and over, and the filaments keep sliding. So a single muscle fiber contracts fully or not at all. How, then, does your bicep produce forces ranging from lifting a feather to hoisting a heavy weight? The first answer is the motor unit. A motor unit is one motor neuron plus every muscle fiber it controls, the smallest chunk of a muscle the nervous system can command. To produce more force, you recruit more motor units. And recruitment follows the size principle. The small motor units are activated first, and larger ones are brought in only as demand rises. This is elegant for 2 reasons. It gives you fine control at low forces. You're adding small increments, and it means the fatigue-resistant units carry the everyday load, with the powerful units held in reserve for when you really need them. Recruitment is one lever for grading force. Stimulus frequency is the other. A single action potential produces a twitch, one brief contraction and relaxation. But if a second stimulus arrives before the muscle has fully relaxed, the second twitch adds on top of the first. That's summation. Increase the frequency further, and the twitches blur into a sustained but still bumpy contraction, unfused tetanus. Push the frequency higher still, and the individual twitches fuse completely into a smooth, maximal, sustained contraction, fused tetanus. This is the strongest force a single fiber can produce. So between recruiting more motor units and firing them faster, the nervous system has 2 independent dials for setting exactly how much force a muscle delivers. There's one more factor that sets how much force a muscle can generate, and it's built into the geometry, the length of the sarcomere before it contracts. Force depends on how much the thick and thin filaments overlap, because overlap determines how many cross bridges can form. There's an optimal length right in the middle, where overlap is ideal. The maximum number of cross bridges can engage. The tension peaks. If the sarcomere starts too short, the filaments are already crowded and collide, and force drops. If it starts too long, the filaments barely overlap, few cross bridges can form, and force falls off the other way. That's why every muscle has a length at which it's strongest, and why your body positions its joints to keep muscles near that sweet spot. Let's close by pulling the 3 muscle types back together. All 3 contract by the same cross-bridge mechanism, but they differ in revealing ways. Skeletal and cardiac muscle are striated, smooth is not. Skeletal is voluntary. Cardiac and smooth are involuntary. The most important difference is the calcium target. In skeletal and cardiac muscle, calcium binds troponin, the mechanism we spent this lecture on. In smooth muscle, there's no troponin. Calcium instead binds calmodulin, which activates a different pathway to switch on contraction. Finally, coupling, skeletal fibers act independently, each under its own neuron, whereas cardiac and smooth cells are joined by gap junctions so they contract as a unit. And cardiac muscle even has its own pacemaker cells. And that ties the whole module together, from a single receptor all the way to a coordinated force-generating tissue. Thank you for your time. See you next time.